Hello and welcome to Talking About My Regeneration, the podcast that's all about stem cell and regenerative medicine and some other stuff too. I'm Kevin McCormack, the Director of Communications at CIRM, California's stem cell agency, and we've got $5.5 billion to change the world. And this podcast will introduce you to some of the people who are going to help us do just that. Enjoy the show. Our first podcast is something rather special. It features a man whose name shall be inscribed on the band-aids on the arms of millions of people who have had their COVID vaccine shot. Dr. Derrick Rossi is the co-founder of Moderna, the company behind one of the most effective vaccines against COVID. He recently sat down for a chat with CIRM's president and CEO, Dr. Maria Millan.
Welcome, everyone. We are so pleased to welcome Dr. Derrick Rossi for this exclusive, inclusive interview brought to you by CIRM. Dr. Rossi is a world-renowned stem cell biologist recognized for multiple scientific discoveries, including mRNA, which many of you are very familiar with, but we'll get to that in a bit. He was recognized for this discovery by Time magazine in 2010 and was one of the people who mattered, along with President Obama, the late Steve Jobs, and LeBron James. He has continued to matter a lot. As the co-founder of Moderna, I'm sure he is extremely proud that that company, as well as others, are giving us a way forward to get to the other side of this unprecedented coronavirus pandemic. We are very excited to hear about his journey in the scientific world and how he brought these discoveries all the way through to lead to this revolution in vaccine development using mRNA. So I also want to say that Dr. Rossi, we're so proud that you were a CIRM scholar early on when the field was just in its infancy. Stem cell field was really, really young when you started. So welcome, Derrick. And of course, the vaccine is top of mind for everyone these days, but we'd like to take a moment to give you the chance to kind of describe your journey, where it started, what it was like to be a postdoc, what brought you to California in the first place. And then I know that you had a long story before then, but what brought you to bring this technology to your own lab at Harvard and then beyond? Thank you.
Sure. Well, first, I just want to say thanks for inviting me to this, and hello, everybody out there in webinar land. So it could be a long winding path, because my path through science is a long one, but I'll quickly, say that I started off in Toronto as an undergrad, and I did a grad work there. I was in Paris for a little while. I ended up doing my PhD in Helsinki, Finland. For some strange reason, took a wrong turn, or the right turn perhaps, at Albuquerque. And I ended up in Stanford to do my postdoctoral research with Irv Weissman in the field of stem cell biology, and particularly its intersection with aging. And that was in the early 2000s, where indeed I was, I think, in the first class of CIRM scholars. The Prop 71 had passed recently, and I applied for one of the scholarships and was granted, which was fantastic. Did 4 years of work at Stanford with Irv, working on stem cells, and then I took my faculty position at Harvard in 2007. And we got inspired to actually, work on mRNA. We're not working on mRNA, although I was classically trained as a molecular biologist, so I know what mRNA is, but hadn't worked with it. In fact, many people hadn't worked for it, which is an interesting part of the story. But what got us into it was actually a discovery in stem cell biology by Shinya Yamanaka. So a Japanese scientist who made the absolute, fantastic discovery that you could introduce 4 genes into a skin cell of a human that you could pull out of my arm or your arm or anybody's arm, and then turn these cells back to a state of, early developmental potency, such that these cells could now turn into any type of cell in the body. And that was a very, very exciting discovery. And indeed, Yamanaka deservedly is awarded, excuse me, the Nobel Prize, and because his, discovery was so fantastic and has so much promise and still does to this day. We, like all other stem cell biologists, were very inspired by this study. So so Yamanaka's approach, though, had a problem, and this is the first reference to viruses now. So he actually used retroviruses, which, by the way, SARS-CoV-2 is a retrovirus, so an RNA genome virus. He used not SARS virus or coronavirus, but a virus to deliver the cargo, which was these 4 genes. And what those RNA viruses do is they integrate them, they turn themselves into DNA, and they integrate into the genome, which is a mutagenic event. And then you're stuck with these, 4 now called Yamanaka factors for the rest of the life of the cell. And that was going to be a problem for clinical translation. So we thought, well, we know that, DNA makes mRNA, makes protein, makes life. Why don't we just cut DNA out of the equation altogether, and we'll just go mRNA makes Yamanaka factors, makes reprogrammed cells, leads to therapies, stem cell therapy. So that was the initial concept. And that was actually derived by a postdoc in the lab, Luigi Warren, who was he was actually the first postdoc in my lab. And we started to, we made textbook mRNA. You can find out what an mRNA looks like. We made beautiful textbook mRNA. But when we introduced it into the cells, we really didn't get the desired response. We got the first gene we made, well, the first sort of test protein was called green fluorescent protein. Its name bespeaks pretty well what it does. It's a jellyfish protein. When you put it into cells and you put the protein as expressed, the cells will glow green under the microscope under a certain wavelength of light. So we got a few green cells in the dish, but we also got a lot of dead cells in the dish. And dead cells in the dish was not what we were trying to achieve. So that could have been the end of the story, but we thought there might be a workaround. And that led us to the work of 2 scientists from the University of Pennsylvania, Kathleen Kiriko and Drew Weissman, who had a few years prior to us beginning our study had published a paper. It was actually relatively unheralded, but they made a really important discovery. And that was that if you used modified building blocks of RNA, they're called nucleosides, modified nucleosides, and they weren't actually making mRNA in that study. They were just sort of trying these modified nucleosides. What would the cell's response? Would it have this sort of antiviral response? By the way, that's why we were killing the cells, because when we were introducing the RNA, the cells were looking at it and it looked like an invading RNA virus, and they were mounting an antiviral response and responding with an appropriate altruistic suicide, killing themselves. So that's why we were killing the cells in the dish, it turns out. Anyhow, the discovery of Kiriko and Weissman suggested that if we use these modified nucleosides, we might be able to get past this antiviral response. And that was really the fundamental sort of thing that enabled our work to proceed. We were able to get mRNA, now modified mRNA, into cells, express any protein that we wanted. We reprogrammed cells. We took those cells and turned them into all kinds of things. But by the time we had sort of made maybe a half dozen or more, dozen or so proteins in the lab, I realized that now we had a really robust technology for making proteins with mRNA, this modified mRNA. And I mentioned it earlier, DNA makes mRNA, makes protein, makes life. Well, DNA mutation makes bad mRNA, makes defective protein, makes disease pathology. And that's pretty much all our genetic disease follows that little mantra. So I envision that if we could make proteins now using this modified mRNA, that we might be able to intervene with genetic disease actually and introduce a copy of a non-mutated copy of an mRNA to somebody with a genetic disease and potentially treat their disease with it. So that was the sort of concept of the founding of Moderna. It was not vaccines, it was not viruses, it was basically genetic disease. But that's what I pitched first to a colleague and then to venture and founded Moderna based on that sort of sequence of events.
I wanted to just kind of highlight and take a moment to really appreciate all that you told us. One of the highlights of this is how the scientific community kind of evolved together. This discovery that you made in your lab and the research being done in University of Pennsylvania, which, by the way, that research, I believe, led to to the Pfizer vaccine. Is that correct? So like in parallel, the Moderna and this other company, BioNTech, that was taken up by Pfizer, those two, the Moderna vaccine and the Pfizer vaccine, are the first out the gate with mRNA technology.
Essentially the same technology. Yeah, Moderna was working on it longer because I founded the company in 2010. And I think actually the good news is it had a lot of success as a biotech company, even in the early days, just raising money and generating excitement and publishing, exciting papers. I think that's kind of what brought BioNTech into the fold as well, and many other companies, actually, to be honest. Many other RNA companies have started, and many are coming. So it is going to be a class of medicine, basically. Right. mRNA is now going to be a class of medicine. We now have 2 emergency approved, mRNA medicines. They happen to be vaccines in this case, but I think it's just the tip of the iceberg, and we're going to see, many more in the years to come. On the other hand, I've been sort of saying that same thing for 12 years now, and it's great to finally see it realized, and obviously very satisfying.
No, it must be an incredible feel. And I think I wanted to emphasize that moment in terms of convergence and this critical mass, which says this is now a field, right? And now mRNA, in terms of vaccine development, it's saying this is now a new era for vaccine development. The first 2 out the gate to FDA approval for emergency use authorization were both novel, never done before mRNA-based technologies, a whole new technology platform. And it's truly incredible, that whole story.
It is, but your point is a really good one, which is, there's such a, scientists always work off the foundation that others before them have laid. And all of these things have to, there's so many different people involved along various stages of the way. And it really is a community. Science is a community where we learn from one another and we build upon one another's work. And that, that is true of every single science paper ever published, but it's particularly true of this study that, our work had 4 bearers before us that did really important work, that had they not done that work, we wouldn't have succeeded. Had we not succeeded, those that came after us wouldn't have succeeded. So we always build upon the shoulders of those that went before us.
Absolutely. And often it's not those who are first or not the first attempt, it's what happens after that. And that's what's so important about the process of science, isn't it? And scientific community and continuing to support science.
Well, also there's a lot to be learned with failure, quite frankly. I mean, it's one of the most informative things that can happen in the lab is you design an experiment and it just doesn't work for whatever reason. And then you figure out, you or somebody else figures out why that is, and an important insight is made. So as I said, when we first did our experiments with mRNA, they failed.
That's right.
They failed. And then we turned to earlier work that then, provided the key for success. And
to that point, you mentioned Dr. Shinya Yamanaka, and he recently said that, somebody had asked what makes it, what makes a scientist successful. And he said one of the things is embracing unexpected or even negative or failed results from what you had originally expected when you designed an experiment, embracing it because they could be the key. And then Jennifer Doudna, recent Nobel laureate, as you know, for CRISPR-Cas9, had said, and also knowing which one of those to follow and how you follow that, isn't it? It's also as a community, because you feed back on each other in terms of informing your path, right?
Yeah.
So it's really an incredible process. And to hear your story really highlights so much of that.
It's really true. It's amazing. Well, I'll tell you that, science, to be a scientist, you have to have a pretty thick skin, because, most of the experiments that you design and do actually don't succeed. They fail. And because you're exploring the unknown and you're making hypotheses and you're trying to design the best experiment possible. So I would say that a very successful scientist has a kind of a hit rate of success of maybe 20% or something like that. So you have to be accustomed to failure, and sometimes that failure tells you nothing, but often that failure will tell you something, provide the insight that allow you to get to the next step. I remember a very and I think Jennifer and Shinya said it exactly right. You have to dig into negative data, because there can be beautiful nuggets in there that inform where you go forward. Don't just throw up your hands and walk away. Oh, God darn, the experiment didn't work, and that's the end of it. It's a time for learning. I remember vividly just a story from Stanford, actually, my time in California, where I was working in the lab of Irv Weissman, a great stem cell scientist and a great human, a friend of mine. But he, one time there was a lab meeting, we had a joint lab meeting, he had many people in the lab, 35, 40 people in the lab. And somebody came into the lab and presented data that showed that a mouse model that the lab had been using for years and years and years and years and published many papers on had a fundamental flaw in it that nobody had recognized before. And Irv's response was, how can we use that? I mean, that was the first thing that came out of his mouth was, not like, oh, son of a gun, we did all these studies based on this model that was incorrect in this fundamental way. No, his first thought was, what can we learn? What can we use this for? And it was really shows you how that mind works.
Yeah, it's incredible. And so it's a really, important lesson for those entering the field, as well as those who are supporters of science, or understanding why it is important to support science, because it's not a linear track oftentimes. It's multiple meandering paths until you find the way, right? And so it seems that the aha moment for you in terms of the power of this mRNA technology was the ability to reproducibly get the proteins expressed. Exactly. And it does have such remarkable potential, and we'll get into that in a little bit. I want to just come back a little bit to the vaccine, because it truly is revolutionary and transformative. The Moderna and Pfizer, both using the mRNA technology, certainly did develop these vaccines at warp speed. When you think about vaccine development, typically 5 years is considered extremely fast. It's typically 10 or even 15 years. So to have these vaccines developed in less than 11 months, I guess, from the time of getting the sequence and developing it, and then putting it through clinical trials of 30,000 or more people, that's an incredible amount to do. But so, I think that one of the things that you pointed out was it was nearly a decade of scientific research that first happened, and then for you and others to recognize its potential so that it can go into a setting where it could be, converted into something that could be a product, right?
Oh, even longer than that. I mean, I would say the origin is the discovery of the DNA code. And, DNA, everybody now knows what DNA is. It holds this, the code of life. Everybody, I think most people on planet Earth know that. They've heard the word DNA. What most people don't know is actually the effector molecules, the stuff that really makes things happen in the cell, are proteins. Proteins have their origin in a cellular compartment called the cytoplasm, yet the DNA lives in the nucleus. So how do you get the code, reading out these wonderful effects? I mean, because DNA is essentially a passive molecule. It contains it's a book, but unless you can read it and translate it, you can't do anything. And you need these proteins to be the worker bees of the cell to do everything. And so, in the 1950s, the discovery was made and the hypothesis was made that there must be an intermediate molecule that takes this information from the nucleus, delivers it to the cytoplasm where the proteins are being synthesized, because otherwise, how on earth do you get those proteins made? DNA is always in the nucleus, protein synthesis always in the cytoplasm. And, so in the 1950s, mRNA was posited as this messenger, and that's its name, actually, the messenger RNA. It's got a great name. It's taking that code from the DNA to the cytoplasm. And that's why I always, like to talk about this story, because everybody knows what DNA is. Almost nobody knows what RNA is, but it's so fundamental to all of life on planet Earth, because, DNA makes mRNA, makes protein, makes life. And that's something that I would say that the general public, I'm hoping, knows, within a year or two, that everybody can say, oh, mRNA, I know what that is. That's the thing that carries the code from the DNA to make the proteins. And, it's relevant for vaccine development, obviously, because what information is that mRNA carrying into the cell? Well, it's carrying the information encoding for the viral spike protein. And it delivers that code on the mRNA into the cell. The cell then does what cells do with mRNA, which is makes proteins out of them. That's what's involved upstream in this vaccine. Makes whatever protein is being encoded by that mRNA, which in this case happens to be the spike protein, which then is recognized by the immune system as a foreign object. Humans don't express the spike protein. That's not in our genome, and our immune systems are very good at our immune systems are built to surveil what is self and what is non-self. And they're very good, the immune system is very good at saying, this is my own material, so I'm not going to react to this, or this is something really weird, we better mount a response to this. It's what all vaccines are based on. And by the way, vaccine takes advantage of the normal process that our bodies do on a daily basis, which is surveilling what is ours, what is self, what is non-self, and mounting reaction to what is non-self. It's amazing to me that people don't actually know what vaccines are. And maybe it's not surprising, because, we get our first vaccines when we're children. You get vaccinated as an infant. You have no capacity to ask questions, or, you don't want to even understand language at that point. And then by the time we're teenagers and we're getting vaccines, then it's just a pain in the neck, because you don't like to get jabbed in the arm to get your shot. But everybody gets them, and they're like, well, not everybody, but many people get them because, they've been proven safe over, millions and millions and millions of people vaccinated. It works. And by the way, they work. They protect against, pathogens. That's the whole objective of a vaccine. So again, when you talk about how did this get to be so quickly, first you have to, just in the mRNA perspective, go back to the discovery of mRNA, which is in the 1950s, because unless we've discovered that that's the intermediate between DNA and protein, we would not have RNA vaccines. And then, the whole group of people working on the precursors to SARS-CoV-2, those that worked on SARS and those that worked on MERS, discovered and really well defined that, this spike protein is really the thing to target, because, so I'll just, hold
No,
it.
I think that you're highlighting some really key points, which is that different fields of research, virology, molecular biology, and then also, we're taking for granted the genes, but actually the Human Genome Project was not that far away. So even knowing how to all the tools and everything to identify what the sequence is that gives rise to the mRNA, they all come together. So that was all in place because of the science that had been happening for decades. And we don't know when we're going to need it. And so they're recruited to do this thing. And what's really incredible, and thank you for pointing out how, to explaining it in a very tangible way, how vaccines work. What's incredible is the efficacy of the mRNA vaccines versus traditional vaccines, because traditional flu vaccines are maybe 30 to 40, 50% effective on any given time, depending on the strain. And we've been developing, we as a community have been receiving and developing flu vaccines for a long, long time. So can you speak to, first of all, 94%, 95% efficacy, both for the Pfizer and the Moderna vaccines in the large analysis that led to the approval. Can you speak to, was this expected? Like, did you expect this level of penetrance and effectiveness? Like, so this actually, yes, we train, our bodies are recognizing the virus presented in, or viral proteins presented in different ways, but this way of presenting it seems to be so powerful. So can you comment on, I know that, there may not be a straight answer, but what are your thoughts on that?
Well, part of the reason that we developed some years better influenza vaccines than others is because we make educated guesses on what strain is coming in the forthcoming year. And sometimes our guesses are better than others. The reason for that is we have to make them so far in advance because it takes so long typically to make vaccines the traditional way that, you make a vaccines, it's predictive of what we believe the strain that's coming this year. And sometimes we get it right and sometimes we get it less right. So that sort of dictates how plus or minus of effective a flu vaccine at the very least is every year. So what's different about mRNA is, again, this, and you pointed it out a moment ago, the fact that we had the sequence, and it was published by the Chinese consortia, this Shanghai consortia in January. We had the sequence of the SARS-CoV-2 virus. So people at the NIH and Moderna, the day that was released, they started thinking, okay, where's the spike protein? There it is. We know what region of the spike protein to go for because we've known from years of looking at the crystal structure of spike protein. And it's an interesting protein because it's in one conformation before it touches and contacts a human cell, and then its conformation changes actually upon engagement of its receptor. But that was known because some people had discovered that. So very good that was all known beforehand. But now, by virtue of the fact that you have the sequence, and you know how this protein works, and you know that that's the one to target because that spike protein is the first thing that engages with the human cell to initiate infection. Actually, it interacts with a receptor on human cells called the ACE2 receptor, and that is the interaction that starts the infectious cycle. So if you could develop antibodies to exactly block that first interaction, then you really have a chance of developing a good vaccine. And you know what the sequence is of that spike protein in that region. So that was what was used to develop the mRNA. And sure enough, the body, very good, developed neutralizing antibodies against this. And the efficacy, though, is extraordinary. But it just, certain pathogens, that's the mechanism that thing gets into the cell. If you block that mechanism, if you're effective at blocking that mechanism, you should be effective at blocking infection. And indeed, that is borne out. Of course, now I'm sure we're going to talk about variants at some point.
Oh, yeah, absolutely. Let's talk about variants. I mean, I think I wanted to just kind of encapsulate the very important points you made, which is that our body is actually the most efficient producer of the right antibodies to respond to the invader. And it's just getting the right way to mount that response. So all the things that we do to vaccinate are different ways to try to kind of, give a warning and say, start your machinery to fight this. But the mRNA is so specific towards something that was identified. And as you, the virologists and all the researchers that had identified this to be the key region had it right. And I think that's amazing how all these things came together. But yes, variants. So given the power of this technology, and as we know, viruses are viruses. That's just how they survive through the history of life, is they mutate, or certain variants, forms of them become dominant so they can keep surviving. So of course, that's a fear, with what could happen, an escape from some of the things we already, that we're putting in place. So can you comment as to the role of this technology in tackling future variants, current variants that are starting to now become more prominent in the community?
Yes, I will. I'm going to just finish the thought from the last question, though. So remind me, and I'll come back to that. But you asked, why is it more efficacious? And I said, well, if you've got an exact sequence, you can make exactly the thing that you want to target. That all is true. But what is also different fundamentally from traditional vaccines, which are usually made with sort of inactivated viruses, you actually take the virus and you inactivate it so it's non-infectious and you give that to people. That's actually all extracellular interaction that's having with the immune system. In contrast, when the mRNA makes the spike protein, it makes it internal to the cell, mounts it on the cell surface. So this might also be part of the reason that you get this more robust, immune response leading to this high efficacy. I just wanted to add that point in because I didn't mention that before. Now, in regards to the variants, so, the viral genome, the spike protein is, part of the viral genome, but there are many other components of the viral genome that work to basically, make sure that this particle is able to replicate in human cells and, go off and be infectious to more cells. Um, so, though the spike protein, as I said, is really the key first step in the infectious cycle, at least for this virus and coronaviruses in general. You really have to have a lot of mutations to change the sort of structure enough to escape the sort of immune response that's already elicited by the current vaccine. That's not to say that it couldn't happen, never say never, but you really, you could mutate the SARS-CoV-2 genome probably 20% or more. Obviously some of those mutations will happen in other genes important for the viral life cycle, but the real key ones for the immune recognition are in the spike protein. And indeed, some of these variants that have emerged are, what do you know, lo and behold, mutations in the spike protein. And yet, though, when the companies, both Pfizer and Moderna just published a paper online the other day, have tested at least the current vaccine's ability to neutralize these variants, so far they do. And so at least the variants that are out there, this B117 and the South African strain are the current vaccine is protective. But never say never in biology. At some point, that spike protein is going to mutate enough that the immune response to the initial inoculation, with the initial sort of knowledge of what the spike protein looked like then versus what it looks like now, after all these mutations happen, that's going to cross a point that it's we're not going to have an effective vaccine. The good news is that with each new strain that comes out, we can very quickly sequence it. We can exactly see what the mutations are and how they change the structure of all the proteins involved, particularly the spike protein here. You can make predictions even, because you know how the antibodies interact with the spike protein. Is this going to interact with those antibodies, yes or no? But at some point it's going to happen. The cool thing about an mRNA technology is, well, guess what? You'll know the new sequence of the new spike protein. You'll just make a new mRNA for that, and you'll start to give that to patients. And at some point, you may notice that every year when we get our flu vaccine, a clinical trial is not run for that. That's because we get our vaccines every year, and vaccines are, they've been given to hundreds of millions of people on the planet. It's really deemed one of the safest possible things you could do for people is give them a vaccine. Certainly the benefits far outweigh the negative side effects. So we had to have phase 1, 2, and 3 for these mRNA virus vaccines because they were new and it's a new technology. Although Moderna and BioNTech had already run a number of clinical trials on other vaccines, also giving confidence that it was safe and effective. Nothing that went through phase 3 though, but small phase 1 and phase 2 trials. But at some point when there's enough trials done with mRNA and vaccines that people say, well, you know what, we know it works every time. We know it's safe every time. The safety profile of by the way, you know, it's going to essentially be the same drug every time. It's just going to be an mRNA with a different sequence, just encoding a different protein.
It is paradigm shifting for sure. It really is, because as you said, all the predictions and checking the chicken, you know, the eggs and all that with traditional vaccine development is you've shifted, it's turned it on its head. And also what's also paradigm shifting is the way that companies had joined forces, and how the all hands on deck approach has led to such a rapid.
Well, that was critical.
You know, and of course the government agencies and the FDA and all of the funders. Thank you so much for that. I wanted to you know, take it back to a public health piece for a bit. So like, let's say you're in an elevator, which many of us are not because we're still in some phase of lockdown.
Stay out of the elevator,
but you are 6 feet apart and with masks, and someone said, oh, you're the famous Dr. Rossi. Can you explain to me briefly, okay, now you've discovered the vaccine, it's all good, right? Like, so why, you know, why are masks and social distancing still important? And where do these antivirals come in? Because they're still developing those. So can you just, like, in an elevator ride, let's say 16 floors, we'll give you a little bit more time, explain kind of in the context of these pieces in terms of tackling the pandemic?
Yeah, well, they're obviously critical. I mean, we know that this virus, you know, transmits and can be passed from person to person through water droplets, you know, that we generate when we're speaking. You know, just speaking generates, you know, tiny little droplets that contain viral particles. That's a deadly combination. When you can pass a virus just by sitting, you know, in proximity to somebody and talking, that's a serious virus. And hence, you know, look at the number of people infected around the planet. That just goes to show you how good of a mechanism that is for those viruses to move around. So masks are key, and social distancing are key. And one of the very popular, I call it popular ways to infect oneself is to, my hands aren't clean, so I'm not going to do it right now. But, you know, you're in an elevator, you press that button, and then, oh, I got an itchy eye. Scratch your eye. Guess what you've just done? You've just taken the virus from that button that you pressed. There's a couple of viral particles there because the person before you touched the button, because we all have to touch that 16th floor button if we're going to the 16th floor. Now you've touched your eye. Very good method for viruses to infect one's body is through the mucosal membranes in the eye. So, you know, diligence is just critical for this. And this is why, you know, from a public health perspective, I mean, just any biologist can give you this lesson that I just gave you, but, you know, all biologists know that. All you have to know is how viruses transmit one another, and then you can take precautions to protect oneself, and not just yourself, but your, you know, those around you. So now what about when the vaccines, when we start all to get vaccinated? You know, for example, you know, when I get my vaccine or you get yours, nobody likes wearing masks. They're a pain in the neck. I wear glasses, and, you know, it's winter out there. So every time I go with a mask, you know, and my glasses, I fog up my glasses and then I can't see what I'm doing. Happens all the time. I leave it all on anyhow because I'm a biologist. But once we get our vaccine, can we take these measures and stop doing them? The answer is no, because unfortunately, you can still get infected when you've got antibodies. The strong likelihood and the evidence is that you won't get sick because your immune system is already primed to respond to your infection, and it gets on top of it very early, therefore preventing you from getting sick. But it doesn't mean that you can't get infected nor transmit to others. So we still have to, until we sort of get the virus under such control that it's not pervasive in the community, we're going to have to be keeping practicing these good health practices. So I don't want everybody to let their guard down once they start to get vaccinated, because it's not, you know, for yourself, in all likelihood, you're not going to get sick. That's what the evidence shows us from the trials. Your immune system would be nice and charged and ready to respond. You probably won't end up in the hospital, but you still have the ability to infect others. So we really have to be careful.
Thank you. And then antivirals are kind of a rescue measure, isn't it? When all these things.
Sure. Well, I mean, there's other things. Even the antibodies which is a biologic, Regeneron and others have made antibodies. These are great. I mean, this is what President Trump got. He got monoclonal antibodies. Is basically artificial immune system given to you. We've just said that antibodies are an important part of blocking the viral sort of life cycle. Well, if you can inject somebody full of antibodies that do that, you don't even have to generate them yourself. But these are not super practical medicines. They're hard to give. They're often IV administered in a hospital setting. They're expensive. But it's great if for really high risk people, I would say that's nursing homes and this type of thing would be a very good thing. If the elderly or the infirm get infected, quickly get some antibodies in them, because they have generally our immune system's potency goes down with aging. And if basically giving antibodies are a good way to give somebody kind of a bit of a crutch of an immune system to help them get over something. And antivirals, similarly, there are different small molecule drugs and others that impact the viral life cycle at various stages. So of course those should be used. Again, that would be post-infection. You'd have an infection. But if you could interfere with small molecules to interfere, we talked about the viral genome has many genes in it, and its life cycle is, complex. It has to make lots of itself, and that involves many different parts of the cell and the machinations of the cell to make a virus. So if you've got a good small molecule that interferes with that, of course it's worth pursuing. But that again is a post-infection. The only sort of prophylactic way to really not get sick in the first place is vaccines.
That's right. Thank you so much. I think that is such a strong message. Another kind of public, up top of mind for the public is that this coronavirus has really kind of highlighted disparities and disproportionately afflicts minority populations and disadvantaged communities. There's multiple prongs to that. I noted that Moderna had published and has on its site the fact that it was built into the trials, specifically to make sure that these populations are proportionally represented, because after all, these are the populations, as you said, for the vaccine to really impact, it needs to be effective and also to be something that's used by the broader population. But so just so what are your thoughts about this topic of, diversity, equity, and inclusion as we're looking at vaccine clinical trials and vaccinations?
Well, I was really happy with Moderna for doing that, because basically you're exactly right. In their 30,000 person phase 3, they tried to model the sort of number of participants from different minority groups and ethnic groups into that 30,000 that would be reflective of the population of the US. It's actually pretty rare for clinical trials. One of the big problems with clinical trials historically is often they're only done on men and not women, which is remarkable. Not vaccines, but other clinical trials. And it's white people. So really, this idea that medicines are not being tested typically on a diversity of individuals is really true, and historically it's true actually. So I think it's really important. I'm really proud of Moderna, although I had no say in, I wasn't asked, but but had I been asked, I would have totally supported that. I think it's great. I think it should also give, by the way, the public, confidence. Again, there's suspicion in some communities of any type of clinical trial, because generally people don't really know what clinical trials are, or how they work and how they're designed. And especially for something that was so quickly brought to market, there's suspicion. I get that question all the time from reporters. Well, there's a lot of suspicion because, or even open forums, well, it was done so fast. We're taking risks here. Actually, the answer is not true at all. Still there was phase 1, 2, and 3, which is true of all clinical studies. What made it so fast was the amount of resource and manpower and money power that was put behind it, and the fact that the companies didn't complete their phase 1 before they started their phase 2 and didn't finish their phase 2 before they started phase 3. They staggered them, they overlapped them, which, by the way, is a really risky thing to do from a business perspective, because you're running a phase 3 trial. You don't even know if your phase 2 trial has worked yet. And so if it doesn't work, your phase 2 trial, you're out of all the money that you spent on your phase 3 trial. It's a very risky thing to do, but it was necessary to get it done, to get, a global pandemic, to provide that light at the end of the tunnel, because ultimately the vaccine is really going to be what pulls us out of this thing. And I think everybody could agree that the last year was really crappy, just for day-to-day life, let alone all those families that, lost loved ones and or their livelihood. I mean, the economic and the social destruction that this pandemic has caused is massive. So, sorry.
Absolutely. And, one of the things is, certainly these were unprecedented. The word unprecedented, I don't think has been used as often as it has been in this past year. Everything's unprecedented. But the good thing is there was also an unprecedented mobilization, unprecedented, technologies, unprecedented response and timelines. I'm just going to shift a little bit and take a question from the audience, if that's okay. And then I, at the end, we're going to wrap it up with some fun questions for you that are circling back to you again. So would it be possible, this is the question, to apply the mRNA delivery method to develop other vaccines, even for those immunogenic viruses? And I think you had spoken about that, but how do you think it would impact for influenza, which is, we talked about that's the one we're most familiar with, but also efficacy is about 30-50%. And this, so is it going to change the paradigm for our common virus that we face every year?
Absolutely. So Moderna has already launched an influenza program. BioNTech, same thing. Other companies will do the same. So in coming years, your influenza vaccine will be an mRNA vaccine, and it will impact in that it will be better, because we'll know the strain. We can wait long enough to know what the prevalent strain is. We can make vaccines that are exactly tailored to the genetic sequence of those particular viruses. So we will have better vaccines each year. I anticipate that essentially the entire vaccine industry will move to mRNA vaccines. Their efficacy is proven, their safety is proven. Ultimately, their cost of manufacturing is cheaper than traditional means of vaccine manufacturing. A little bit expensive now because it's, setting up the manufacturing infrastructure and the raw materials that are needed and this and that. But those, as those manufacturing facilities are developed, and they are, by the way, going to go up around the planet, you're going to see mRNA manufacturing facilities in multiple places. Because, by the way, guess what? The next time we have a pandemic, because there will be a next time, that's for sure. Viruses are really, almost the perfect evolutionary units. They will find a way to infect us large scale again. It will happen. But this year it was unprecedented. Within a year, we, Moderna and BioNTech, got their drugs approved. And then within this year, they said now they'll make 1 billion doses each. And the reason that it's taking a year is because the infrastructure for that manufacturing is, Moderna was smart. It made it invested a lot of money in a manufacturing facility, actually in Norwood, Mass, which is 20 minutes from where I live here. They did that already a couple of years ago, anticipating, clinical grade mRNA. But now these manufacturing facilities will go up around the world. So think about it. The next time we get a sort of a global pandemic, maybe we don't have to run 3-phase trials again. Maybe we could only run a quick one, or maybe by that point, mRNA vaccines will be so commonplace that we'll just say, well, you don't even need to run a clinical trial because these things work and they're safe every time. It's a possibility. But now, instead of producing 1 billion doses in a year, oh, well, we've got manufacturing facilities. We'll access the one in Ireland and the one in Israel and the one in Canada, and we'll have 1 billion doses this month.
Yeah, it's really phenomenal. I mean, just what's in it, and also the FDA in partnership with the scientific community and the developers, and looking at the different ways of still ensuring safety and making sure it's gone through the rigorous process, but being able to accelerate that process.
Do you know what's really important about that? Actually, I was really impressed with the FDA. They actually live streamed the committee meetings for the emergency approval of these drugs. That is never done. So you talk about transparency to the public. You could actually, and I did, I spent the day in front of the YouTube watching these, or not the YouTube, but the stream, my computer, watching these meetings of the committees assembled to assess the data and to ask the questions and go over everything. And it was all available to the public. And that is really cool transparency. I think it's really important for this. That is not normally done for drug development. These commissions and these committees are not done in the open like that. So it's actually been an incredibly transparent process, more so than ever. And I sometimes get reporters say, well, it was so non-transparent and this and that. I said, well, actually, it actually wasn't. It was really transparent. You could go and read all that data. It's all there. Oh, you can watch those meetings. I'm assuming, I watched them live, but I'm assuming that there's a link that you can still watch them. That's
incredible. And yeah, so truly incredible. I mean, when we say partnership, it's every sector of the scientific policy, regulatory and citizens all banding together on this. And here's another question, and it leads to, what are the other applications for this technology beyond vaccines? So here's the question. Based on the success we see with mRNA vaccine for COVID virus spike protein, I understand there's an effort to use mRNA to encode for myelin protein that can be used as a vaccine for multiple sclerosis. Is Moderna working on this, or is anybody that you know of working on this, and how promising might it be?
Well, I know the study that's in reference to. It was actually a study done by the two scientists out of BioNTech, the sort of husband and wife team. I've got to say that's a really, really early study in a mouse model of MS. It's called the MOG-YAE model. It's actually not a very good model for real MS. So it's early, very, very, very early days for that. Because the problem is, what the antigen is, the thing that's MS, autoimmune disease. So the mouse model, you actually put an antigen, what the antigen is, and they sort of, they protect it against that antigen. But when people develop MS, and there's 1 million Americans that have MS. I'm working with a company, Convello. We work on MS drugs. So I know this field pretty well. They don't know what the antigen is, autoantigens, and then there's antigen spreading as well. So it's a much more complicated problem with patient MS than it is with this mouse model, which is not a very good model. So I'm sorry to be not that hopeful in that particular question. Moreover, when you first start a program and you publish a mouse study on it, and like I said, this particular mouse model is not particularly great, it'll be, many years in development before a medicine gets to patients. You've got to go through phase 1, you've got to go through phase 2, you've got to go through phase 3. Now, we've just seen that with a global pandemic going on, you can do that in a year. That is typically not what happens though, because as I said, the trials will go one after another, they won't stagger. And the amount of resource involved and the scale of the effort for any other thing other than a pandemic is just not as massive. It's not to say that MS is not a hugely important disease. As I said, 1 million Americans just Americans suffer from MS. Very debilitating disease, terrible disease. But it's going to be a while. Now, do I think that mRNA will have a role at some point? Possibly, but it's going to be down the road. But I do believe that mRNA therapies will be in many other settings, and indeed both BioNTech and Moderna have multiple different clinical programs at various stages of development, either phase 1, phase 2, to get different programs eventually approved using this mRNA technology.
Absolutely. So, I mean, it's a powerful technology, and sometimes you just need those other key pieces of data and information that to determine how to use the technology. Isn't that the case? So there is more science to be done in terms of elucidating some of the basic, science knowledge we know from MS, even to maybe that to determine whether that could be applicable. Here's another question. And I don't know. So the question is, I am interested in the outlook for treatment of lingering effects of COVID. Would the Moderna vaccine have any impact in the case of a patient who has had a bout of COVID in terms of easing or preventing the long-term effects, meaning a therapeutic, I guess, potential application versus a vaccine application to the vaccine? Is that something that's ever been considered?
I'm not sure I understand. So somebody's been infected with COVID, they've already mounted a response, and now give them the COVID vaccine. Yeah, well, there hasn't been studies done on that, number one. So to really answer the question, you'd have to do the clinical study, if I understand that that's what the question.
Yeah, that is the question. And so the studies haven't been done. Theoretically, if the reason for the sequelae or the long-term effects is there's this continued mounted response to this lingering signal, right? That potentially, but it's not something, that's all theoretical. All right, so here's another question, and then we're gonna wrap it up with our closing. Can you comment on some of the, it says anaphylactic shocks that have been reported after second dose delivery? I don't know if this is with Moderna per se, but maybe with vaccine.
There's been some with Pfizer and vaccine, Moderna as well. Yeah, I mean, it's something that's quite rare, and I think that the quote unquote culprit is not actually the mRNA or even the lipid particle that's carried in, but some other components of sort of, consider benign components, something called PEG is in this. What I would say about that, though, is that everybody that has had these responses are somebody that really has a history of allergies, and pretty much they all have EpiPens with them, and they take their EpiPen and then it resolves very quickly, and or where they get vaccinated, they've got EpiPens around, and so these things are resolved very quickly. They're extremely rare. They didn't even rear their heads in the in a significant way in the 30,000 trial done by Moderna or Pfizer, but now as hundreds of thousands, even millions of people are being vaccinated, these rare events are occurring. The good news is, like I said, they're totally manageable and resolvable. What I would always say, what I like to add to that is, though, because then some people say, well, why should I risk that? Why would I get a vaccine if I have this 1 in a million chance of getting, an anaphylactic shock? Well, I would say if you don't get your vaccine, you've got a much higher risk, about a 1.5% risk, somewhere in that range, of getting COVID and dying from COVID. Mm-hmm. That's a much higher risk rate than 1 in a million. And I don't know, by the way, I don't know if it's 1 in a million.
No, it's absolutely right. It's like with any medical intervention, it's always a risk benefit analysis, or that one has to do, because nothing is 0% risk. Absolutely. That's exactly right.
That's the point.
So I wanted to just, first of all, thank you again, Derrick, for such an amazing interview. It was really great, and so much information packed into this wonderful 1 hour meeting together. Thank you so much for being with us, and for joining us at CIRM and with our audience.
That's our show for today. I hope you enjoyed it, and thanks for listening. I'm Kevin McCormack, and we'll be back soon with another edition of Talking About My Regeneration. See you then.